So we have seen how meiosis occurs, okay? And we have seen that it occurs in 2 stages. So now let's see how actually it occurs, the stages. So we saw that, yeah, there are 2 stages, there are 2 successive divisions, or a cell undergoes 2 successive divisions, that is meiosis 1. But even under meiosis 1, the phases are the same. The cell can undergo, not can, but the cell undergoes phase 1, metaphase 1, anaphase 1, and telophase 1. And we need to know actually how a cell appears under all these phases. Can we mute? That's good. How can you, if I give you a cell, for example, I draw a cell and I draw it like this. Maybe it's just a sketch. Okay. And I say, which phase is this? Will you be able to tell the phase? Or you just watch it? Or if I give you a microscope and I show you cells, and you're good enough, you see some things, will you be able to tell that this cell is under this phase? So that if you can do that, then that will be the science. Then another phase or another stage is meiosis 2. And with meiosis 2, the same thing. It has prophase 2, metaphase 2, anaphase 2, and finally telophase 2. And we shall tell, if I give you a cell, you need to identify that this cell is in anaphase 2 of meiosis, or this cell is in anaphase 1 of meiosis. Okay, so are you ready with quiz? I guess so. So now let's see what prophase 1 is about. How does a cell appear when it is under prophase 1? And actually, let me tell you this. This prophase 1 is actually the phase that is kinda complex, kinda complicated, kinda good as far as meiosis is concerned. Now let's see how a cell appears. Like any other prophase, that's when chromosomes will appear. But unlike the prophase of mitosis, where just chromosomes are just scattered within, okay? The chromosomes are just scattered. Here, the chromosomes will be seen in pairs, okay? And these pairs, they are pairs of homologous chromosomes, okay? The chromosomes are seen in pairs, and these pairs are pairs of homologous chromosomes. That's all, guys. Let's proceed. We shall see. Then they will pair up in the process known as synapsis, and that pair of homologous chromosomes that will be formed is known as a bivalent. Okay, so in case you observe a cell under a microscope and you see those chromosome pairs, you see bivalents. Sometimes the bivalents might be crossed. They might cross like this. We are going to see what it is and the significance of this. Then you know that that cell is under prophase 1. Don't just say prophase. Don't just say prophase. If you say prophase, then you are referring to prophase of mitosis. But if you say prophase one, then that's when you are specific, man. That's when we shall know that, yeah, this person knows what they are talking about. And when we are marking, we shall give you such a good tick. Okay? Brilliant. Now, this is good. This is key. Now, the non-sister chromatids, if you remember, I said if this is a chromosome, just chap, and this is its chromosome pair, and the two chromosomes are homologous. This chromatid here and this chromatid here, the same applies to this and this. They are referred to as non-sister chromatids of homologous chromosomes. Now, in some cases or some chromosomes, these non-sister chromatids, they may wrap against each other. Like this, if you do this, and then I do this, then this one may wrap against the other one. Okay? And they form something like a coil. And then when they wrap against each other, they may end up crossing each other. Of course, you cannot wrap and fail to cross each other. Now that point of intersection, that point of union here, okay, is known as a chiasma. Chiasma, okay, or is known as chiasmata, okay. It's okay. It's okay. Yeah. Somebody wanted something, but yeah, it's okay. So that point, if you have this chromosome and you have another chromosome, and the two chromosomes, the two homologous chromosomes, meet each other. Now this point here, where the two non-sister chromatids meet each other, is known as the chiasmata. Okay, I think this is the plural form. And then when it's one, it is the chiasma. Now don't confuse it with a centromere. You know, students use from past experience, students they confuse this point with the centromere. Is this that point where the two chromatids meet each other on a chromosome or join each other on a chromosome, is known as a centromere. But now when the two non-sister chromatids, like how you see this, fuse or they coil against each other and join and cross each other, that point where they cross each other from is known as a chiasmata. Now you should be wondering what actually happens when that chiasmata or when that chiasma is formed, or when the chiasmata is formed. You can fact check that. The plural form, the chiasmata, and then the singular. You can fact check that. Okay? Now what happens after you formed the chiasmata? These segments, I think I've ever illustrated that. I think I've ever illustrated that for you. The segments, this one, remember this is for this chromosome, it is for this chromatid, and then this one, it is for this chromatid. Now this is going to break at this point, and this one also breaks at this point. Now when this breaks, it will join this chromatid. The same applies to this, it will also break and joins this chromatid. And the same will happen on these very chromatids up. This one also break and joins this, and this one also break and joins this. Now that process of breaking and joining these segments is known as the crossing over. Okay, and I'll set a question. Okay, let me not cheat for you. I'm going to set it. Okay, that process of breaking, first of all, you first join, and then you break these terminal segments, these ones, and then you rejoin them on the segments of other chromatids. Is known as the crossing over. And the crossing over occurs in meiosis one, but to be specific, in prophase one. Now, you are not just exchanging segments. No, you are not just exchanging segments. Remember, these chromosomes carry DNA. Okay? They carry DNA. Now, the moment you break this segment from here, you are breaking, the whole of this is DNA. The same applies to this. The whole of this is DNA. So if you break this and you add it here, and you break this and add it here, it means there will be change in the DNA. You completely form new combinations. Okay? And those new combinations, they will be manifested in the phenotype as variations. And that's when you actually see organisms belonging to the same species, but the two organisms are totally different. We studied variations, you remember, and we saw one of the causes of variations is crossing. So that crossing over, it occurs in prophase one of meiosis one of meiosis. That's brilliant. That's brilliant. So this causes crossing over, which involves exchange of chromatid material between chromosomes from the parents. This leads to combinations of genes when or in sex cells. And I've already explained that. So this is the appearance of a cell. If I give you such a cell, sometimes you might have the spindle fibers forming. You may have that aster, this the pair of a centriole together with the radiating spindle fibers, these microtubules. That's called an aster. But that is all minor. Because we saw it is the same for mitosis also. Here, the main thing which identifies that this cell is in prophase one is you look at the arrangement of these chromosomes, how these chromosomes are. Do you see this? This is one chromosome and this is another chromosome. And the two chromosomes have paired up to form what we call a bivariate. Okay, you can see. Okay, and this point where you can see how they are even exchanging. This one, it is coming here. This one is also here. The same applies to this, the same applies to this. And so that point where the two chromatids meet from is known as the chiasmata. The chiasmata, this is plural. If it's one, it is chiasma. And I told you to fact check that. Okay? But it is 99% correct. But that 1% is also possible. So you fact check that. So, and that's what I'll give you. That's what I'll give you. I'll say, identify which stage of cell division is this cell. And then I give you objectives. So you guys should be in position to answer any question. So I hope prophase one is okay. In case there are questions, please feel free to ask. Okay, now before metaphase one, I want you guys to answer this question. The question says, which of the following which one of the following phases does crossing over occur during meiosis? It seems some cool question. Somebody has posted the answer already. Okay, that's good. We have part A. We have metaphase 1. We have B. We have prophase. We have C. We have anaphase 1. And D, we have prophase 1. So what's your answer? So what's your answer? Somebody has already posted the answer. Okay. Before I even finish the question. So the answer is C, and a good percentage passed the question. However, some few. Yeah. So the answer here is prophase 1. Why? Because we just saw crossing over, and we saw that crossing over, it's where you're going to exchange the segments. And we saw it occurs in prophase 1 of meiosis 1. So that's simple. That's how simple that question was. And always expect such question. Okay? Expect such question as far as cell division is concerned. Okay, that's good. Let's see what occurs during metaphase. Let's actually see what metaphase is. Now, like any other metaphase, like we saw in metaphase of mitosis, that involves arrangement of chromatids on the spindle equator. Now, here, they are pairs of homologous chromosomes that arrange themselves on the metaphase plate on the spindle equator, such that during anaphase, when they are separating, each chromosome of the homologous chromosomes of the bivalent goes to the other, to the opposite poles. I think we have the drawing here. Yes, this is the drawing. So this is it. I want you to have a look at this, and then I'll also take you back to where we studied mitosis. I want you to have a look at this. Can you please have a look at this? You see how these chromosomes are arranged? That's brilliant. Now, let me take you to where we studied mitosis, and you also appreciate, you see how the chromatids are going to be arranged. So this is the metaphase of mitosis. Do you now clearly see the difference? Do you see the difference? Do you notice the difference? Exactly. Now, with this one, the chromosomes will arrange on the metaphase plate in a way that when this spindle fiber pulls towards this side, and this one pulls towards this side, the chromosome is going to separate. The centromere is going to separate, and the two chromatids will move to opposite poles. And this is metaphase of mitosis. Now the metaphase of meiosis, let me take you there. Let me take you there so that you can. Yes, now this is metaphase of meiosis one. So do you now appreciate the difference? Now the metaphase of meiosis one, these bivalents, these homologous chromosomes, they arrange themselves on the metaphase plate in a way that if this spindle fiber pulls towards this side, and this one also pulls towards this side, then the two homologous chromosomes, they will separate and each chromosome goes to the opposite cell or to the opposite pole. If you can understand that, then you'll be good to go. So in meiosis one, there are chromosomes which separate. There are homologous chromosomes which separate. But in mitosis, it is actually the centromere that separates, and the two sister chromatids migrate opposite poles. If you can understand that, if you really understand that, then you'll be good to go. Can I receive some questions?